In the last lesson, we talked about triacylglycerols, which stored energy by placing 3 fatty acids on a glycerol backbone, resulting in a very large nonpolar molecule that has to be stored in special adipose cells. Many of our membrane lipids are nearly identical with one small change. One of the fatty acids is replaced by a phosphoester. The phosphates in phosphoesters are always negatively charged, meaning they're very polar, and the alcohols that are also part of that phosphoester generally come with positively charged ammoniums, as you can see at the bottom, so this changes the physical properties of the lipid drastically. Again, since one of these 3 chains is now significantly polar. We call these molecules with a glycerol backbone, 2 fatty acids, and a polar phosphoester bond, a glycero for the glycerol, phospho for the phosphorus, lipid, glycerophospholipid. Some of your membrane lipids will use a different backbone called sphingosine, but have the same basic structure. 2 long nonpolar chains, one from the sphingosine and another from a fatty acid, and a third short and very polar chain. We call this class of compounds sphingophospholipids, sphingo for the sphingosine, phospho for the phosphate and the alcohol, and lipids because it's a lipid. Whether the backbone molecule is glycerol or sphingosine, the structure of the membrane lipid will have the same 2 important features: the nonpolar fatty acids and a very polar phosphoester. We're looking at glycerophospholipid here. While it's convenient to draw all 3 chains coming off the same side of glycerol, as shown in the top left, the actual structure will have the polar phosphoester and the alcohol portion flipped to the opposite side of the nonpolar fatty acids, as shown in the top right. This gives 2 distinct portions or regions of the molecule, which we call the polar head and the nonpolar tails. The polar head can interact well with water, which is also polar, while the tails do not interact well with water at all, and seek to interact only with each other. You should know the name of this type of compound, a glycerophospholipid. You should also know that it's a membrane lipid, and you should know that its basic structure has a glycerol backbone to 2 fatty acids and also to a polar phosphoester. You should also know which parts interact with water. The polar head will interact with water while the tails will not. Sphingosine can replace the glycerol and a fatty acid together and create a sphingophospholipid. Structurally, not much has changed. There's still a polar head from the phosphate and the alcohol, and there are still 2 nonpolar tails. Just one is from the sphingosine now instead of from a fatty acid. The fatty acid tail is actually connected as an amide in this case instead of an ester, which is the case for the glycerophospholipids. It doesn't make any difference structurally though. It still connects as a long nonpolar tail. You should know this name, sphingophospholipid. You should know that it's a membrane lipid, and you should know that its basic structure has a sphingosine backbone connected also to a fatty acid and a polar phosphoester. You should also, just like glycerophospholipids, know that the polar head, the phosphate and alcohol portion, interacts with water, while the nonpolar tails will stay away from water. Your cell membrane also has a third type of membrane lipid, also based on sphingosine, called a Sphingo Glycolipid. Here the sphingo still refers to sphingosine, glyco refers to a carbohydrate, and then lipid refers to the fact that it is a lipid. The overall structure is the same though, since the sphingosine and the fatty acid are still the same as a sphingophospholipid, and we're just swapping out that polar phosphoester for a polar carbohydrate region instead. As this is the polar part of the Sphingo Glycolipid, this carbohydrate rather, that carbohydrate is typically shown on the outside surface of a membrane, where it can interact with water. This is an important way for cells to recognize each other. The types of Sphingo Glycolipids present on a blood cell, for example, actually determine blood type. It shouldn't be a big surprise that the major role of a membrane lipid is making up our cell membrane. A cell membrane's biological role is to keep the insides in and the outsides out. One of the main ways that we drive operations in our cells, and therefore in our bodies, is by differences in concentrations. If our membranes can't keep things separate, it's difficult for our cells, and therefore our bodies, to gain or use any energy. Membrane lipids help to do this by separating the waters above from the waters below, or rather the waters inside a cell, which are polar, from the waters outside a cell, which are also polar. Membrane lipids create a lipid bilayer, which is really just a fancy way of saying a layer of lipids 2 molecules thick. The inside of that layer is all nonpolar. It's all the fatty acids from those lipids and the sphingosine tails. And the outside is all polar. It's all the phosphates and the alcohols, so that can interact with water. Polar things usually can't get into the cell on their own, since they're too polar to pass through the nonpolar center, so they get stuck on the outside interacting with the polar heads. They never want to enter the nonpolar center. If we have a nonpolar thing, there's a good chance that it's not easily dissolved to begin with, but if it is able to move through the polar portion of intercellular space, it will get stuck in the middle of the membrane in many cases, if it can get there at all, because the middle of the membrane is nonpolar and it would rather be there. If the membrane fatty acids are too strongly interacting, the membranes start to be more rigid, they turn more solid, and generally that's not good for a cell. One way to make membranes more fluid is to increase the amount of double bonds in fatty acids, because those unsaturated fatty acids won't interact nearly as strongly. Another way this occurs is with cholesterol. Cholesterol is a membrane lipid, but it just sits in the middle of the membrane, in that nonpolar region by all the fatty acid tails, and it disrupts the interactions between those fatty acids. Looking at what's present in this molecule, is this molecule likely to be soluble in water? The only polar thing on this whole molecule is a single alcohol. And so we have, well upwards of 20 carbons. And this is a very nonpolar molecule overall. That means it's insoluble in water. And it also means that it's very difficult for it to interact with water. Cholesterol needs to be carried by something polar when it moves through our bloodstream, which is mostly water. There are two carriers that move cholesterol around in our bodies. The first is low-density lipoprotein, or LDL. LDL carries cholesterol out from the liver into tissues. This is the type that eventually allows it to accumulate in your bloodstream, the bad cholesterol. High-density lipoprotein, or LDL, carries cholesterol back from the tissues to the liver, which is generally a good thing. Your body actually needs both, though, which is why most blood work also reports a ratio of high-density lipoprotein, HDL, to low-density lipoprotein, LDL. In either case, the cholesterol itself doesn't actually get absorbed as HDL or LDL, since the protein carrier actually gets digested into amino acids. So you can't just eat more good cholesterol to make the balance of those better. Exercise usually increases the portion of your cholesterol that's high-density lipoprotein. It's usually a good thing towards healthy cholesterol levels. And as many of you know, controlling cholesterol is also the goal of a number of significant drug classes that you'll talk about in pharmacology if you're a nursing major. Our membrane doesn't just include membrane lipids. It also includes proteins to help things move in or out of the cell. There are three ways that things can move across the cell membrane. The first is passive transport, which is another name for diffusion. Only small molecules can move appreciably in this way. Generally, small, nonpolar molecules like oxygen or carbon dioxide can move effectively across the cell membrane passively, and water to a pretty small extent. Not much else can move this way because if it's polar, it wants to stay on either side of the membrane. If it's nonpolar, it wants to stay stuck in the middle. So think of passive transport like the wall of your house. Air and moisture can get through some, but it's slow and it's difficult. If there's more air pressure outside, air will eventually move into your house. If there's more inside, air moves out of your house. Passive transport is the same way. Things can move, but they don't move particularly efficiently across the membrane as is. Most things are allowed into the cell by facilitated transport. Think of this more like a door or a window in your house, which is conveniently also how most things get in or out of your house. In cell membranes, there are special doors or gates to allow through molecules. They're specific to certain types of particles. So there's a different gate for the sodium ion than there is for the calcium ion. Gates work like doors. When they're open, things move through them based on concentration, always from high concentrations to low, just like with passive transport. So if you're baking cookies inside and you open a door, the strong cookie smell moves out from the kitchen where it's stronger and more concentrated and gradually dissipates, so you smell it weaker outdoors as the cookie smell continues to move outdoors. And it works the other way too. If they're doing sewer work outside your house, the strong smell moves into your house where the smell is initially less concentrated. Similarly, if you have a lot of sodium outside a cell, a gate will let some of that into the cell. If you have too much carbon dioxide, a gate might let some out, but it's always moving from high concentration to lower concentration. We can also pump things into your house or out of your house. Your air conditioner actually pumps heat out of your house to the outside, even though there's often more heat outside. But it costs energy to do that. That's the same with a membrane pump. We can always move glucose into a cell, even if the concentration is much higher in the cell than outside. But we can only do that if we use a pump. And we always are going to spend energy in order to do that. So a pump is a lot like a gate, except we can move in the opposite direction and we spend energy to do that. The next class of lipids are emulsifying lipids. An emulsifier is a molecule that helps something that's generally not soluble become more soluble. Here, emulsifying lipids help dissolve nonpolar lipids in water so they can be taken up by your cells. The most important class of emulsifying lipids are bile acids. So bile acids are an example of emulsifying lipids. Bile acids, like the cholic acids in the top left, and taurocholic acid and glycolic acid, which are shown on the bottom right, are all derived from cholesterol, which is highlighted, has a blue region highlighted in the very top left. In every one of these molecules, you can see the same four-ring structure. These cholesterol derivatives are synthesized in your liver. So this is actually one of the endpoints for the high-density lipoprotein cholesterol, the HDL cholesterol. These bile acids represent a much more significant portion of the cholesterol and related compounds in your intestine, again underscoring that cholesterol in your diet is actually only a minor factor in controlling your cholesterol levels. Again, all the bile acids are derived from cholesterol. They're made from cholesterol, but they're all modified with an ionic group to make them more polar on one end. You can see that highlighted in kind of orangish, reddish, whatever you call that color, or in green. That helps to make sure that that end, the polar ionic end, can interact with water, and the end that has all the rings, the four-ring steroid structure, can interact with the less polar compounds that your body wants to take up. The next class of compounds are messenger lipids. Messenger lipids are examples of hormones, that is, compounds that send signals around your bodies. Hormones can include a wide range of molecules, including inorganic molecules, but most hormones are lipids, and most lipid hormones are steroids. Steroids have the same core structure as cholesterol, those four rings we call the steroid structure, and cholesterol is the starting material to make them. There are two main groups of steroid hormones. One group is the sex hormones. These include estrogens, female sex hormones, and androgens, male sex hormones. Even though these are typically categorized exclusively, all men have some level of estrogens, and all women have some level of androgens. In fact, androgens are used to make estrogens, so the balance between those is actually what's more important. Progestins are pregnancy hormones. You might be able to hear the same root in progestin as in gestation. They are not just responsible for maintaining pregnancy, but also for menstrual cycle regulation. I also have anabolic steroids listed as a fourth class, but they're actually a type of androgens. Anabolic steroids regulate anabolic pathways. Those are the parts of our metabolism that build up larger molecules. They're typically used medically to restore protein structure, but they're also used unwisely in athletics to build up protein structure as well. Because anabolic steroids fall under the umbrella of sex hormones, they often have unintended results, both for men and women, because it disrupts the balance of androgens to estrogens. A second class of steroids are the adrenocorticoid steroids, or just corticoid steroids for short. They're made in the adrenal gland, and the most common or most well-known corticoid steroid is cortisol, the stress hormone. There are two major types of corticoid steroids. One is mineral corticoids. Mineral corticoids regulate the minerals sodium and potassium, while glucocorticoids regulate glucose. They also have implications in reducing inflammation and suppressing immune function. Dexamethasone, which is used to treat COVID patients that are on ventilators and has many other uses as well, is an example of a glucocorticoid. Not all messenger lipids are hormones. Fatty acids can actually be modified to become short-range messengers, which we call eicosanoids. They serve as messengers within a specific tissue, but not across the whole body like hormones. So eicosanoids are just another example of a messenger lipid that's really only for a small range of tissues. Our final type of lipids are protective lipids. Protective lipids are waxes, like earwax or the wax that covers a fruit, and they always consist of a fatty acid that's super long, tied to an alcohol that's super long, through an ester. They form strong interactions with themselves because they're super long and straight, which means there's lots of London interactions. This creates a very good physical barrier, and since it's nonpolar, they don't let very much through. Not water, not any of the pesticides that can be dissolved in water, not any of the bacteria that need water to live, and so they're really good at protecting. Thanks, protective lipids.